In this video, we're going to look at the action potentials in cardiac muscle. This is Chapter 20, Part 7. So the action potential that's initiated by the sinoatrial node travels along the conduction system that we talked about in the last video and spreads out to excite the atrial and ventricular muscle fibers. And these working atrial and ventricular muscle fibers are called contractile fibers. So these are the fibers that are actually going to contract when the heart pumps. Now, an action potential occurs in a contractile fiber in this way. We have this diagram that shows us how an action potential propagates in one of these muscle fibers. So the first step that we have is this really rapid depolarization. So this is very different than those autorhythmic fibers. So these contractile fibers do have a stable resting membrane potential that's really kind of here around -90 millivolts. Now, when a contractile fiber is brought up to threshold by an action potential from the fibers around it, its voltage-gated sodium channels are going to open really quickly. So these voltage-gated fast sodium channels are going to open really quick. Now these channels are actually a lot quicker than normal voltage-gated sodium channels, like the type that we see in neurons. So we do have this really quick inflow of sodium down its electrochemical gradient, and this produces this really rapid depolarization. Now within just a few milliseconds, those fast sodium channels are going to automatically inactivate, and that sodium inflow is going to decrease. So this entire length is 0.3 seconds. So from like here to here, I could use a different color here, from about here to here, where we have that really rapid depolarization and then subsequent inactivation of those sodium channels is just milliseconds. Now after those sodium channels are inactivated, we kind of hit this next phase of an action potential. So this is called the plateau, and this is a period of maintained depolarization. So this looks a little bit different than the action potentials we see in neurons, where we're going to have kind of this up to threshold, increase in voltage, shoot up and straight back down, and then this kind of repolarization back up to resting membrane potential, right? This is kind of the normal action potential that we see in neurons. Now this picture looks quite different because we have this sustained depolarization, or this plateau phase. Now this plateau phase is partly due to this opening of voltage-gated slow calcium channels. And this is in the sarcolemma of the muscle fibers. Now when these channels open, calcium ions are going to rush into the cell from the interstitial fluid, and we're going to have a really big increase of sodium concentration within the cytosol that ultimately triggers contraction. We also have many different types of voltage-gated potassium channels within the sarcolemma of a contractile fiber. And so depolarization, this sustained plateau of depolarization, is really sustained because we have slow calcium influx, and it just balances that outflow of potassium. So instead of all the potassium rushing out and us getting this really sharp downward turn of the action potential, we're going to kind of balance out that charge differential. And so even though there is some potassium channels open and we have those positive charges leaving the cell, trying to bring the cell back down to a lower membrane potential, we have these calcium channels that are balancing that out and maintaining that plateau, that depolarization plateau. Now, the final step of an action potential in a contractile fiber is going to be the repolarization phase. So the recovery of the resting membrane potential is during the repolarization phase of a cardiac action potential. This looks pretty well close to other excitable cells like neurons. So we have this delay, which is really quite long in cardiac muscle because we have this plateau, but this is where we have more potassium channels open. So here we're going to have kind of more of those potassium channels opening, and some of those calcium channels are going to start closing. So we have those closure of calcium channels, and we have additional voltage gated potassium channels are opening. So then that extra outflow of potassium is going to restore that negative 90 millivolts negative resting potential. Now we've seen this picture before when we were talking about metabolism of skeletal muscle. Now, cardiac muscle is a little bit different than skeletal muscle because cardiac muscle produces very little of the ATP it needs by anaerobic cellular respiration. So we can almost entirely cross this off the list. Instead, it relies almost exclusively on aerobic respiration from all those large, many mitochondria. So we're almost going to completely rely on aerobic respiration. There also, just like skeletal muscle though, cardiac muscle does produce some ATP from creatine phosphate. So very little from creatine phosphate, but mainly we're going to be looking at aerobic respiration as the main means of producing ATP. As action potentials propagate through the heart, they're going to generate electrical currents that can be detected from the surface of the body. So an electrocardiogram, which is abbreviated either EKG or ECG, these are interchangeable, this is a recording of those electrical signals that are put off by the heart. Now on a typical EKG, we have 3 very clear, recognizable waves that appear for every heartbeat. Now the first one is called a P wave, and it's quite a small little hill or upward deflection on the chart here, and this represents atrial depolarization. And this atrial depolarization spreads from the sinoatrial node through contractile fibers in both atria. Now the next wave is called the QRS complex. So QRS complex. So this begins as a downward deflection. So you can see this right here is a little downward deflection of the Q, and then it continues as this really large upward triangular wave and then ends in another small kind of downward wave here. So this is going to be pretty much this whole section here is our QRS complex. Now this complex represents very rapid ventricular depolarization. So this is as the action potential spreads through the ventricular contractile fibers. So this is ventricular depolarization, QRS complex, and then over here we have P wave, which is atrial depolarization. Okay, now we have this third and last wave is this larger dome-shaped little hill, which is called the T wave. So it's going to be this section right here. And this represents ventricular repolarization. So this repolarization occurs as the ventricles are starting to relax. Now, on a typical EKG, we also want to look at measuring the time spans between waves. So these are called intervals. So the first interval we're going to look at is called the PQ interval. And this is the time from the beginning of the P wave. So right here, beginning of the P wave, to the beginning of the QRS complex. And this represents the conduction time from the beginning of atrial excitation to the beginning of ventricular excitation. So we're kind of looking at the beginning of atrial excitation to the beginning of ventricular excitation. That's the PQ interval. Next we have the ST segment. And this begins at the end of the S wave here, and it begins at the end of the S wave, and it ends at the beginning of the T wave. So this section right here, and this represents the time when the ventricular contractile fibers are depolarized. This is during the plateau phase of the action potential. So this is kind of during the plateau, if you can read that. And then the last interval we're going to talk about is the QT interval. So these are just the 3 intervals that you need to know. The QT interval starts from the beginning of the QRS complex. So right here. And then we're going to go all the way to the end of the T wave. And this is the time from the beginning of ventricular depolarization to the end of ventricular repolarization. So this is, we're going to look at from the beginning of the initial excitation of the ventricles to the end of the ventricles relaxing. So that's what that segment is. So the atria and ventricles are going to depolarize and then contract at different times, and this is because the conduction system routes those cardiac action potentials along very specific pathways. Now we'll learn a couple different terms here. So first off, the term systole. I guess we could write it. Systole refers to the phase of contraction. And then relaxation is termed diastole. So we have an EKG, and these waves are going to actually predict the timing of atrial and ventricular systole and diastole. So in this picture on the right here, we have a typical heartbeat. And we're going to start off with the cardiac action potential reaching the SA node. So this is where we just have the action potential coming here, and it propagates throughout the atrial muscle and down to the AV node. And as the atrial contractile fibers depolarize, we have the P wave form in the ECG or EKG. So this is just the action potential, the depolarization of those contractile fibers in the atria. Now, after the P wave begins, this is when the atria contract. So they're going to contract after they depolarize. So this is where we have atrial systole. This is kind of after the P wave in the EKG. Now remember, conduction of the action potential is going to slow down at the AV node, and that's because the fibers in the atrioventricular node are much smaller in diameter, and they have a lot fewer gap junctions than in the SA node. And this results in about a 0.1 second delay. And this time delay is really important because it gives the atria time to contract, and it really gives the ventricles time to fill up with blood before they contract, because we want to allow enough time for enough blood to go into the ventricles so that we get a full pumping action with as much blood as we can out of those ventricles. So we're going to have the action potential is then going to rapidly go again from the AV bundle, and that depolarization is going to progress down the interventricular septum and then upward from the apex. So we're going to come down to the apex, where the apex is down here, and we're going to come around outward from the endocardial surface, and this depolarization of these cells in the ventricles is going to produce the QRS complex. Now, at the same time that this is happening, we're also having atrial repolarization occur. So the atria are repolarizing, they're relaxing, but we don't really see that on the EKG because these very large ventricles and the depolarization of these large ventricles kind of masks or overshadows that repolarization of the atria. Next, we have contraction of these ventricular contractile fibers or ventricular systole, and this begins really shortly after that QRS complex is on the EKG, and it continues during the ST segment. So this is when ventricular systole is occurring. Now, contraction is going to proceed from the apex toward the base of the heart, and so this is how blood is kind of squeezed upward toward the semilunar valves. Next, we have repolarization of the ventricular contractile fibers, and it's going to begin at the apex of the heart and spread throughout the ventricular myocardium. So this is where we have repolarization. And this produces the T wave in the EKG. So this is the actual repolarization of those fibers. And then shortly after the T wave begins, we're going to have the ventricles start to relax. They're going to go through diastole. So pretty shortly after ventricular repolarization is complete and the ventricular contractile fibers are relaxed. So as you can see, we also have seconds on the bottom here, and this entire process from the initial depolarization of the atrial contractile fibers to the last step of ventricular dystole only takes about 0.6 seconds, so it is a very quick process. And that's going to be the end of Chapter 20, Part 7.